Good morning, good afternoon, and good evening. Thank you for joining today's BioCelerate webinar on leveraging CDISC data sets for study analysis. The BioCelerate team on the phone today, I'll introduce are Todd Page, Director of Toxicology at Eli Lilly, Bill Hauser, Principal Scientist at BMS and SEND CDISC Team Leader, Mark Carfagna, Senior Research Advisor of Toxicology at Eli Lilly, and Kelsey Jakey, Director of the Portfolio at BioCelerate and Principal Consultant at PA Consulting. My name is Gloria McHugh, and I'll be your Transcelerate webinar host today. Before we jump into the content, I'd like to cover some logistics for today's webinar. As you heard when you dialed in, all participants will be muted for today's call. You should be able to use either your phone or a computer to listen to the audio. You can reduce the control panel for a better view of the presentation by clicking on the orange and white arrow in the top right of your pane, and that'll move the control panel off to the side. We'll be using questions to submit questions today during the presentation. So use your questions panel, type your question into the panel, and then be sure to click send in order to submit your question to the presenters. If you have any technical issues or would like to report any feedback, please send an email to events@transceleratebiopharmainc.com. We'll also be using live polling today as part of our webinar to make sure it's interactive and facilitate sharing information. Your participation is completely voluntary and all responses will be anonymous. Answers will only be shared as a percentage of respondents. And we may be using results from today's live polls at some of our upcoming events to be hosted by BioCelerate or published online. So at this time, I would like to launch a poll to test out the functionality and also to make sure that everyone can hear us. So you should be able to select your favorite color. Please select your favorite color by voting in the poll. And I'll leave that open for a few more seconds. Great. Thank you, everyone. It looks like we've got about 90% of folks on the phone have voted already today. So thank you very much for your participation. And I'll go ahead and share the results of that poll. So it looks like 65% of today's callers have blue as their favorite color. So thank you very much for testing out our functionality, and it sounds like everyone can hear me okay. I'd also like to cover some ground rules very quickly to make sure that the discussion is helpful and we can answer your questions as well as we can. Participation, again, is voluntary, as is using Transcelerate or BioCelerate assets and tools. And you don't have to identify what company you work for. Things that we would ask you not to discuss would be commercial relationships, your long-term development plans, or anything related to costs. And specific topics that we cannot answer questions about are related to commercial relationships, costs of using or implementing BioCelerate assets or tools, or which member companies are using those assets or tools. We have a variety of representatives at today's webinar today. In terms of types of organizations, we have about half of the folks who registered for today's call are representing research sponsors and biopharma companies, but we also have a good variety of folks representing technical vendors and other types of service providers and vendors, as well as health authorities and regulatory agencies, industry groups and consortiums, as well as CROs and contract labs. In terms of geography, the bulk of the folks who registered for today's session are representing North America, but we also have a smaller percentage representing Europe, Asia, and Central and South America. So thank you very much to everyone for dialing in today and making time for today's call. And with that, I'll hand it over to Kelsey Jakey, Director of the Portfolio for BioCelerate, to walk us through the agenda and take us from here. Kelsey?
All right, thank you, Gloria, and thank you everyone for joining today. I'll walk us through the agenda quickly here. We will give a quick overview of Transcelerate and BioCelerate for those who are less familiar with the organization, and then I will pass it over to our expert presenters to discuss an overview of the SEND harmonization initiative for study analysis, as well as a discussion on some of the sources of variability in SEND data sets that this team is exploring. And then we'll also take an opportunity to let everyone on the phone know how you can get involved in next steps. And we will save some time at the end for Q&A. So just as we go through the content, keep in mind that you can ask us questions, again, through that questions panel, as Gloria mentioned. So if you do have a question that comes up while we're presenting, feel free to type that in right away as opposed to waiting toward the end. Okay, next slide. So overview of Transcelerate and Biocelerate. Who is Transcelerate? We are an organization that was created back in 2012 as a not-for-profit entity, really with the express purpose of fostering collaboration in the biopharmaceutical industry. It currently consists of 19 member companies who have come together to deliver a pretty wide variety of initiatives across clinical R&D as well as pharmacovigilance, all with a vision of accelerating and simplifying R&D. Some of the strategic goals that Transcelerate has chosen to focus on over the years include things like harmonizing process, enabling information or data sharing, and also improving the experience that patients, sites, and CROs have partnering with sponsors in clinical research, as well as enhancing some sponsor-level efficiencies. But beyond Transcelerate, if we move to the next slide, something that several of the member companies of Transcelerate discovered back in 2016 is the opportunity to expand the portfolio and use the same model of collaboration that they had been using in the non-clinical space. And so that is when 5 of our member companies decided to form a separate subsidiary to focus specifically on non-clinical research. And so it does follow the same model as Transcelerate of launching and identifying and developing initiatives aimed at making non-clinical R&D more efficient. And of course, the member companies that are currently involved in Biocelerate are the ones that you see here: Boehringer Ingelheim, Bristol Myers Squibb, Novo Nordisk, Lilly, and Shionogi. Next slide. So what does Biocelerate focus on? Well, right now we're focused on 3 initiatives aimed at making non-clinical R&D more efficient. The first and the one that perhaps most people are familiar with is our toxicology and background control data sharing initiative. This was designed to empower our participating companies to make more data-driven decision-making on compound progression. And what this platform enables is the sharing of both toxicology and background control data sharing within the Datacelerate platform so that participating companies can search for that data, share that data, and also visualize and perform analysis on that data. The second major initiative that we're focused on is common templates for non-clinical studies. This is an initiative that's really focused on making lives easier for the sponsors and CROs and others partnering together on non-clinical research. One of the very first deliverables that we just launched last year is the protocol template deliverable, focusing on first-in-human enabling 28-day repeat dose studies. And so we're in the early stages of the design period for that deliverable, and we're right now in the middle of a public consultation period to get feedback on that deliverable. And finally, the third initiative that we're focused on, and the one that we'll focus mainly today, is the SEND Implementation for Study Analysis initiative. I won't go into too much detail here because that is the focus of our expert presenters, but this initiative is really focused on looking at the CDISC standard and understanding opportunities for greater harmonization of that standard as it applies to cross-study analysis. So more on that in just a minute. Next slide. One thing I wanted to pause on here before we moved on is the work that we have done in data sharing through the Datacelerate platform. I think it's instructive to share that both the Biocelerate and the Transcelerate teams are currently or will be contributing data to this platform across both the non-clinical and the clinical realm. And so as we talk about particularly harmonization of the CDISC standard, I think it's important to note that this team has quite a bit of lessons learned and experiences to share through the development of that data sharing platform. Right now, we have about 35 non-clinical studies submitted in this platform, and they are steadily increasing. and about 75% of those studies are in the SEND format. And so obviously a lot of the lessons learned that this team has accrued over time in developing this platform and contributing data in the SEND standard is definitely instructive for the SEND harmonization initiative. All right, without further ado, I will now pass it over to Todd Page to give us an overview of the SEND initiative.
Thank you very much, Kelsey. So if we can go to the next slide, please, Gloria. Thank you. So I'm sure that most of you, if not all of you on this call, are obviously very familiar with the CDISC SEND data format. I'm sure many of you work with SEND data on a daily basis, and so we don't need to kind of go into the background, but I think it is helpful to just think about where we were, where we are today, and hopefully where we want to go tomorrow. And so if you think from a historical perspective, right, all of our toxicology data used to essentially be locked in a text-based study report. And if you wanted to look up a value from an individual animal, it was a matter of turning to a certain page in a report, or as we got more sophisticated with PDFs, perhaps you could search on an animal number and you would pull up a variety of entries for that animal, and you could kind of click through until you got to the data of interest. Maybe you could then sort of manually build some type of a graphical representation of that data or a chart, and use that to sort of understand what's going on in your study. With the advent, right, it was a tremendous opportunity. It was a giant leap forward in terms of how we can analyze data on a single study. No longer was the data locked, in this text-based format, but now we're able to very quickly, right, manipulate, variables over time, over animal group. We're able to pull out, individual animal data, essentially at the click of a mouse. And so this represented a giant leap forward in terms of analyzing data from a single study, and it's really helped us to understand, at a very in-depth level, what's going on in a study. And I don't think our consortium is unique in this. Obviously, when we started to understand what the opportunity SEND brought to us, we started to think about other ways that this data could be used, right? So if you go to the next slide, Gloria. So not only within a study, right, but think about the opportunities now that exist when we look across studies. So, if you do a set of studies, right, to support a given molecule, you do maybe a 1 month, a 3 month, and then you go to chronic studies, you can, very easily, right, say, okay, what is the progression of a particular tox finding, with time, over the course of all those studies. If you get enough of those examples, maybe you get an understanding of, how a particular finding is likely to progress if it is observed at 1 month. What does margin erosion look like? It could also be used to understand off-target toxicity, right? So if you have multiple compounds, perhaps that bind the same target, you can quickly see, okay, do those compounds have a similar toxicity profile? If they do, that's a pretty good hint that that's an on-target toxicity, and you can start to think about how you can search, huge databases of information to get that. Another use case that we see is understanding the effects of vehicles, right? So, and from a 3R's perspective, right, we never would want to compromise a tox study because of an unanticipated finding with a vehicle. Sometimes it's hard to pull out, data from vehicles, and so if you have all of this data in SEND format and you have a giant database of GLP tox studies, you simply can type in a vehicle of interest, and it would spit out, here's the tox data from this in a rat or in a monkey or what have you. Another use case is thinking about rare and incidental findings, right? How many times do we have something pop up on a study, and maybe it's one animal in a mid dose, and it's uncertain relation to treatment. And we do this, many of us with our CRO partners all the time, but wouldn't it be great if we also had our own database where we could say, okay, what is the incidence of that particular finding, in control animals across, again, a vast library of studies. And then thinking even further along, thinking about developing QSAR, relationships to toxicology data. So that's obviously farther down the road, but that's kind of the next step, is integrating structure now, into some of these tox results. And so what you can see here is that the advent of SEND data actually allows us to do all of these things. But there is a problem, right? There's always sort of a but with some of this. And Gloria, if you go to the next slide. So the but is that as it stands right now, and as I mentioned kind of in that initial slide, the SEND standard was designed to allow visualization of a single study, and it does a great job at that. But if you look at sort of what the current situation is, we're trying to do a graphical representation of this. From any given source or two sources, there can be subtle differences in terms of what a SEND data set looks like. And so both of those study types can be SEND compliant. That's not what we're talking about. But there can still be variability in there that precludes cross-study analysis. And so, if you look at some of those studies from source A, they're not going to match source B, right? Because there's some subtle differences. The overall, architecture is the same, but perhaps in a couple of domains, there are some differences. Maybe there's a difference, in the way units are expressed, or maybe, how you're pulling out, animal birth dates. Bill is going to get into where we see some of the significant sources of variability. But what this does then is, if you want to do cross-study analysis, right now the way people are doing this is that it's really sort of a manual process where people are going in and, kind of manually harmonizing or doing other sort of, data scrubs and data fixes to get data to match up when it's put into a warehouse. And so what our group wants to do is we want to help the industry get to a place where we have this harmonized implementation of SEND specifically focused on allowing cross-study analysis. And so we think if we're able to sort of define, some of these best practices that allow this, and then obviously share them with the industry and have widespread implementation, it's essentially going to unlock all of this data, and it's going to enable all of us to get at our use cases. And so, if we're able to do that, and if you go to the next slide, Gloria, what this does, we think that this is actually a win-win, for multiple stakeholders. So this is actually, for me, it's one of the reasons why this is a fun project to be on. And so, if you think about it from a pharma perspective, a sponsor perspective, we've talked about the use cases we want to enable. Well, what do those use cases do, right? It allows us to design better studies. It allows us to have greater confidence when we advance a compound, speeds data review process. From an FDA or from any health authority perspective, and we actually have some feedback, from health authorities that this is a problem for them, that they actually can't, do some of this cross-study analysis because of some of the heterogeneity in these data package. And so it would make them, able to, review the data more quickly, more rapid study evaluations, perhaps, more speedier decisions, when you file an IND or even when you, submit an NDA. From a CDISC perspective, we think, again, we're just one more voice to give some input to them on how they can further develop and advance the SEND standard. And so we obviously want to be very collaborative with them as well. From a tech and a vendor perspective, if we can get some widespread adoption, some of these recommendations, and it's going to be, more consistent across all of the customer base, and we think it can also streamline some of the analytics that they develop. And from a CRO perspective, if all of your clients are asking for, the SEND data sets to be developed in a similar way, we think it's going to increase efficiency on that end as well. And ultimately, the biggest winner in this is obviously the patients. right? So getting our medicines to market more quickly is going to get them available to patients, and that's what we're obviously all here for. So with that, if we go to the next slide, Gloria, we have an audience poll. And the first poll is, so I talked about some of those use cases before, and we want to hear from you. What do you think is most valuable for use case? And so one of them is the understanding the tox profile of all studies per compound. So this is kind of that margin progression over time. Understanding off-target or trends in a particular class or mechanism of action of compounds. Having a database of vehicle effects. Understanding frequency of rare or incidental findings, or again, ultimately, being able to match up the SEND data with QSAR, or even, I guess we don't have another. I guess we don't have another one here, but so pick your top of those 5 options.
Thanks, everyone. It looks like about 50% of the people on the phone have voted so far, so we'll leave that open for a few more seconds. I know there's a lot to read there. All right. Thank you very much. We'll go ahead and close that, and I'll share the results.
So again, the top vote getter there was the tox profile of all studies per compound. So I thank you all for participating. I would say we didn't have another option there, but if there is a use case perhaps that we haven't highlighted here that you think is important, feel free to share that in the questions. And so if you go to the next slide, Gloria, with that, I'm going to turn it over to my colleague Bill Hauser, and he's going to talk about where some of this variability resides in SEND data sets.
Thank you, Todd. And let's go on to the next slide. Yes, as we begin thinking about how to use SEND data sets for cross-study analysis, we have decided that it would make sense to categorize the domains into 3 different categories. Because when beginning to do an analysis, you first need to identify which studies are applicable for the analysis you're trying to do. And a lot of the information that is needed to decide which studies to include are in the trial summary domain. And so we consider that to be the category A, or the highest priority domain to ensure the harmonization is established. Because if you can't collect the studies together that you want for your analysis, you can't really even do anything. So we think of the trial summary as sort of a card catalog to your library of studies in your warehouse. And just like a card catalog in a library allows you to determine which books you will want to find, the trial summary domain enables that kind of looking for which studies to retrieve. Then once you've selected your studies to analyze, then there is the category B domains. This is the next set that you would be looking at. These are the domains that describe the study structure. So you can determine which animals received no dose, which received high dose, which ones were recovery, those sorts of things that are important to understand the context of any measurement. So we consider those as the next priority. If we find a variability in one of these domains, that's the next, that impedes our ability to do cross-study analysis, then those are the next high priority. And then, of course, the data is ultimately the most important, because if we don't have useful data, the previous things are useless. So category C is next. But if we can't understand the context of the data, nor understand that it was a study we wanted to look at, that data doesn't really give us any value. So we are classifying the data domains as category C. So going on to the next slide, we can consider the use cases that, that Todd mentioned. There's the first one of understanding the toxicology profile for all studies on a particular compound, and seeing that is the highest priority for what the poll indicated just a few moments ago. This is a key area that we want to make sure works well. So if you're trying to do this, you first want to select all the compounds or all the studies that were conducted on a particular compound. So you'll go to the category A domain to see the compound identifier and select all those that have the matching compound identifier. One of the challenges, though, is that there isn't a universal compound identifier that exists at the time studies are conducted. So there's that's an area for standards improvement. We have thought of some potential solutions, but nevertheless, there is a bit of a challenge there. Within one company, it's your own company's policies to be able to use the same identifier, and so that may work for many cases. Then once you've selected those studies, we're on to determining which animal received how much. And we look at category B, the study design domains for that. And then we can look at the toxicology profile through the data domains. Another challenge with this use case is that some of the controlled terminology has some very near synonyms, like the dose frequency. If you look at the controlled terminology, there's actually 5 different ways to say something that is very similar to once weekly dosing. There's 1 time per week, 4 times per month, every week, and Q7D. It's great that there is the controlled terminology that we can, if you're looking for a study with that kind of dosing frequency, you can look and see what else is similar and ensure that all of those terms are included in your search. But some of the other fields aren't controlled as much and makes it more difficult. And then there's the actual dosing is available in the EX records. So you can look at the planned dosing information, or you can look at the actual dosing information, giving a variety of different ways that this analysis could be done. Another challenge is that while the standard uses ISO 8601 for the date formats, date and time, a lot of the analysis tools don't know how to interpret that. So that's more of an implementation challenge of how can we use ISO 8601 dates in graphs and calculations of time differences. And one of the sources of some of this variability is in the wording in the protocol. If the protocol says that the dosing would be 1 time per week, then it's very likely that the SEND data set would end up using 1 time per week. If instead the protocol says every week, then that's likely to be the dosing frequency term that gets into the SEND data sets. So that's part of the reason that we have this sister project on the Common Protocol template to help. We can work together with that team and propose wording standardization that will improve the SEND harmonization. The next use case we want to look at, it's on the next slide, is for off-target toxicity. And we go through the same kind of process here. We can look in the category A domains, the trial summary, for the pharmacological target. And then once we have the target, studies that worked on that target, we can look in the category B domains to understand how much each animal received, and then in the category C to see the effects. Some of the challenges here are that the trial summary parameter for pharmacological class is not very clear as to what should be placed in it. The Veterans Dictionary is a list of terms that can be used for studies that have already been or compounds that have already been approved, and so sometimes that's useful guidance for what to supply, but in other cases, if you're working on a new class, there may not be any standard term for it yet. So to help mitigate that, we propose to CDISC several planned pharmacological target terms, and they are now available in SEND using controlled terminology from March 2018 and more recent. So that may help to some extent. So that's some of the challenges here with looking at off-target and on-target toxicities. The next use case, number 3, so the next slide, and it's is looking at the vehicles that might be used across studies. Here, this can be helpful when planning the next study to understand the effects of the vehicle on previous studies. The challenge in SEND, though, is that there is no guidance it's just a free text field. And looking at how it has been used on some studies, we have 3 examples shown here. It might be some proprietary name of a solvent saying that it was in water and was used. No indication of how much, what the ratio was with water. Another case, you might have a list of short terms that might be understood or may not be understood, like PEG 400, PG, TPGS, citric acid. You may not know if it's a liquid or a powder, and you certainly don't know how much of any one of those. Some of them give very clear concentrations of each using percent by weight. Some might have percent by volume without any indication of the temperature, making the repeatability less possible. So again, the wording that's in the protocol often drives the wording that is used for the vehicle in SEND. So there's an opportunity to work with the protocol, standardizing the protocol to help more standardization in SEND. There's also a possibility for greater structures in SEND to hold the vehicle information. The next use case, number 4, is for understanding the frequency of rare and incidental findings in controls. And so here, the key thing to look at is which animals were the vehicles or were in the control group. And the biggest challenge there is that the standard doesn't give us a standardized way of determining whether a control group is a positive control or a negative control. And obviously, if you include some positive control data animals into your pool that you think is all a vehicle control, you end up with some findings that really shouldn't be there. So that's one of the challenges there. The next slide talks about the variability drivers. We all have that collect data. They have standard operating procedures that guide our processes. And then we also have the study protocol or the study plan. And from the plan, following the line to the left, the plan specifies what should be collected on the study. And so in the laboratory information management system, the LIMS data collection platform, we then collect the data. And then from the LIMS system, we produce the SEND data sets. And depending on the capability of the LIMS system and how that vendor stores the data and the tools we use for extracting the data from the LIMS system and putting it into SEND, it results in some different flavors and different ways in which the SEND data sets are presented. So that's some of the variabilities that creep into the process. Also, as I've mentioned before, looking at the line between the study protocol down to the right toward the SEND data sets, the wording in the protocol can also influence how SEND is prepared. And each organization has different understandings of the SEND standard or different preferences of how different fields should be populated. So there's that variability that drives the results as well. And of course, the capabilities and preferences in each of these three areas, the LIMS, the protocol, and our SEND preferences, also influence how the SOPs are prepared and defined. So these all is a sort of a web of interconnected components that result in some variabilities. The next slide summarizes these. The variability in SEND causes analysis challenges in all those use cases we looked at. And some of those variabilities are from the SOPs that are defined differently across the various companies. Perhaps the ratio of how much is specified in SOPs versus how much is specified in the protocol or study plan. That what is actually in the protocols and plans. The LIMS capabilities, the packages that are used, how that is different between each place where data is collected. And then there's the flexibility within the standard. Each of these variations influence how SEND is prepared. So I'm curious how you view these variations and which ones do you think are more significant and more important than the others. So would you please vote?
Thanks, everyone. So far, we've got about 60% of the people on the phone have voted. So we'll leave that open for a few more seconds. All right, great. We got about 87% of the folks on the phone voted. So I'll go ahead and share the results.
Very good. So we see that the highest is the protocol and study plan variations, followed by the SEND standard flexibility, and then the lower importance of LIMS systems and SOPs. Thank you. Another question for you is when looking at the variability that you've seen, have you noticed a variation between geographic regions? Do you experience varying interpretations of SEND implementation across different geographies?
Thanks, everyone. We're up to about 70% of folks on the phone have voted so far, so thank you for responding. We'll leave that open for a few more seconds. And I'll go ahead and close that and share the results.
Okay, so kind of a pretty close to equal split between the three options of having no visibility to it, to that there isn't a lot of variability across the studies or that the geographies or that there is. Thank you. All right, so with that, I'll turn it over to Mark Carfagna for solutioning roadmap and the next steps. Mark?
All right, thanks, Bill. So the next step, and after identifying these sources of variability, is really working on how we can work together to work on these challenges that we've identified. And again, as Todd pointed out earlier, this is a multi-stakeholder endeavor that requires input from a large number of individuals. And at this point, with the webinar, we're at the point where we want to, across the stakeholders, identify and validate the challenges. This is our take on the issues and the challenges, and we really want to get feedback from other organizations on how they see those challenges and if there's different challenges that need to be considered. The drivers of variability, again, same situation as we've done our analysis and looked at it, but we're sure there's other opinions out there on what those sources are, and would love to hear those. And then once we get into the solution phase and start thinking about how to put this into practice, we're interested in best practices and learnings. If other organizations have the ability to answer some of these use cases that we brought up, what those best practices are. Prioritizing, based on the poll, it seems like a lot of people want to look at all the data from one compound. I'm sure there's other cases that would be used also, and we want to prioritize those and develop our systems to address those in a prioritized way. And then finally, we're interested in how we can work together as all stakeholders to have input on the guidance and how the guidance is used, and further develop the SEND standard to facilitate these types of analyses. Next slide, Gloria. So this process that we're talking about, this collaborative process, we've broken it down into 4 stages. We're about in the middle of trying to understand challenges and drivers of variability, and also to prioritize focus areas. The second half of this is again, getting stakeholder feedback and understanding the different views on these compared to what we've come up with at this point. Once we get through that part of the evaluation, we start to identify solution options. These, I'm sure there will be a large number of them, and we need to sort and prioritize those. And then we get into the final process of drafting proposals or guides, and then finalizing the proposal or guide with the groups that need to perform that activity. So we're at the beginning of the journey at this point, and again, we're looking for feedback and input from all the stakeholders involved. Next slide. So how can you get involved? Obviously, we have a website. The Transcelerate website is available, has lots of information. We have some important information on there from the public consultation memo, which has some details on what I just talked about from a stakeholder perspective, and what some of the thinking is, and views moving forward. You can sign up for updates, and you can also register for upcoming events. So it's a good source of knowledge for the BioCellerate program, and contains information that may be helpful in your use of it. Next slide. Those of you attending the Society of Toxicology meeting next week, there's 2 things happening there that, if you're interested, we'd like to see you at. First is we're presenting a poster on Tuesday, March 12th, and this is an outline of our proposal to better harvest the value from SEND data. So we'd love to have people come by and love to discuss the proposal and hear your input on that. And then also Wednesday, the following day, March 13th, at the SOT meeting at the Hilton Baltimore, we're having a luncheon and a workshop to discuss details of the current proposal and again get input, have roundtable discussions with people from multiple stakeholders. So again, if you're at the SOT meeting and you're interested in participating in any one of those, especially the workshop, please go to our events page and sign up for that. Next slide. Okay, I'll turn it over to Kelsey for questions and answers.
All right, thank you, Mark. And before I do that, I just one clarification. I recognize that not everyone on the phone today will have the opportunity to attend the SOT conference next week in person. So I do just want to make everyone aware that we do plan on publishing the minutes from that meeting to our website so that others have an opportunity to engage with some of the things that were discussed. And of course, we'll continue to update the site with any other additional opportunities for you to engage with us, either one-on-one or in a live roundtable style forum later on this year. All right, so just moving on. Oh, go ahead.
Yeah, I'd like to jump in and mention that workshop is, it doesn't to attend it, you don't have to be registered as an SOT attendee. So if you want to come just for the workshop, that's certainly a possibility without having to register for the SOT conference.
Yeah, thank you, Bill, for that clarification. All right, so I'll move on to the questions then. We did get a few questions, and actually I'm going to start with a more specific and targeted one that came up earlier in the conversation. This one is directed toward Bill. As you were talking through the different categories of spend, and in particular the card catalog, the design, the data domain, there was a question that came up around how we would categorize the DS domain. Bill, would you mind answering that question?
You said DS. Is that, if I understood correctly, the
DS.
Disposition?
Correct.
Oh,
I believe it was disposition. Yep.
It seems pretty critical to understand the phase of the life of the animal to understand whether it was recovery or not. I'm not sure if we've fully agreed as to where it belongs. It would certainly not be category A, the study catalog, but I could see arguments for it to be in either B or C. Sorry for not giving a definitive answer, but that's where we are.
Thank you, Bill. And I think more of a broader question that was actually submitted prior to the start of the webinar that I'd love to hear from Todd, Bill, and Mark on, because I think gets toward more of a philosophical question on what will it take from a standards development perspective to make future standards more consistently implemented in the industry. Is it about more required variables? Is it about more controlled terminology? What are your thoughts on that? And I'll pass it to Todd first.
Thanks, Kelsey. So I thought this was a great question, actually, and as a group, we kind of had a chance to all, think about this a little bit. For me, though, I guess it kind of goes back to, what I mentioned earlier on the call, in that, you need to, when you're thinking about standards, you need to think about what the end is. What's the intent of these standards? What are the standards intended to enable? And with SEND, they were intended to enable, visualization and analysis of a single study. And so from that perspective, I think they very much were fit for purpose, right? You never want to be overly prescriptive, because then that obviously, is a barrier to implementation, and it makes it more challenging. And so it really needs to be fit for purpose and fit for the purpose of what you want to do with this data at the end of the day. So I don't think the answer is, more variables or more controlled terminology, and I don't think by any means is this, any sort of a deficiency with, the CDISC send standard as it was intended to be implemented for the use case that they had at the time. I think it's just that we've identified a new use case, and so to implement our use case, right, we need to perhaps be a little more prescriptive than was originally intended, and I think that's very reasonable approach to take with this. So I don't know if I specifically answered it, but I guess that's kind of how I think of it. Is the standards need to be kind of fit for the purpose of what you intend to do with the data.
Mm-hmm. Yeah, and as Todd mentioned, that the challenge in developing a standard for this kind of data is that on one hand, you want to ensure that there is adequate flexibility so that as science evolves, the standard can continue to be used as new variations of how people conduct the studies. If it's important to be able to convey that, it's useful to have enough flexibility for those sorts of things. And on the flip side, you want to have enough standardization across the studies that we can consistently analyze the data in a consistent way. And so I see that as we're evolving the use of SEND, we'll be probably introducing ideas of how additional standardization will be helpful to satisfy these new use cases. And when we all agree on what those areas are, then CDISC will be able to see what changes to make to facilitate these new use cases.
Yeah. So, and the one thing I wanted to add was that we don't see this as necessarily adding additional variables or controlled terminology to the standards. What we're really looking for is how to leverage the flexibility that's built into the current standards to be able to do these cross-study analyses that we talked about today. So it's really trying to shape the current standards and try to have a harmonized way of using them to enable these types of analyses.
Thank you, Mark. And that actually is a great lead into the next question, which is another philosophical one, and one that I know the BioCelerate team has already debated quite a bit. Wondering if Bill could answer this one. So the question was, how many problems with cross-study analysis should be solved by the individual data sets versus having ontologies and algorithms in the data warehouse to join study data for a specific analysis? Bill, what are your thoughts on that?
Yes, we've debated that question quite a bit. There are two camps of thought. One is, let's improve the structures and implement standardization at the creation point, and that will then simplify our analysis downstream. The other thought is that if there are algorithmic ways to extract the data from the SEND data sets with the variability that they are currently being prepared, then we can rather quickly implement those standardized analysis solutions to be able to mine our existing data. And then there would be no need for the additional changes that will cause additional cost and effort to implement across the industry. And if we have found a solution like that and yet introduce some standardization, then that introduces yet another variation that these algorithms would have to deal with in order to analyze all the data that has been collected from the beginning of send through whatever time we're at in the future. So you can certainly join us and add your voice and thoughts to which way is best to guide our solutions. So consider joining the discussion.
Thank you, Bill. So the next question here refers to harmonizing on vehicle descriptions. What are your thoughts on how we can harmonize on vehicle descriptions? For example, controlled terminology may obviously be needed. How can we answer the problem of harmonizing on vehicles? Mark or Bill?
I don't know, Mark, do you want to start or should I?
Yeah, I can start. I mean, I think the first thing that we can do fairly easily is to be diligent about using concentrations and amounts that are in vehicles. I think those are that's where a lot of confusion comes in. And the other is the use of proprietary names and considering other ways to represent those vehicles and making them unique enough that we can search across studies.
Right, and we would want to recommend a harmonized approach that is not difficult for any of us to implement. So it will require a discussion with all the stakeholders to understand what is what kind of delimiter is going to be easy to use between the components, what way to represent the concentrations, so that we can all implement it with the least effort and get the greatest value from it.
All right, thank you both. The next question here is more from an implementation standpoint. We already have a CDISC send IG guidance document. Now the question is, can we update this existing guide rather than coming with additional guidance to follow? It may be challenging for industry to comply with various sets of additional guidance. Todd, what are your thoughts on how we can work with the existing send IG?
Sorry, I was on mute.
Are you on mute?
Yeah. Can you repeat the first part of that, Kelsey?
Yeah, so just the acknowledgement that we already have a CDISC send IG and guidance within that. And so I think the question is, how do we leverage this existing guide as opposed to adding additional guidance on top of it?
So, and it's a great question. It's something we've talked about as a group. We're certainly not in the standard setting business, within Biocelerate here. That's not what we want to do. That's not our core purpose by any means. I think what we're proposing is something that complements the CDISC send implementation guide. And so I think that if people want to implement this, it will require some additional work. And I think that there's an opportunity to collaborate with CDISC on some of these recommendations. I guess as I've kind of viewed it, and Bill, feel free to chime in. Bill's very heavily involved in CDISC. I look at our group as maybe being a little bit out in front of where CDISC is as a standard setting organization. They're going to be probably a little more, watching and observing. It takes them a little more time to respond. But I certainly hope that, we can have a very constructive dialogue with them, and that they can, sort of understand what some of our recommendations are, and that eventually, those will get, I hope, implemented into the actual, CDISC send IG. But in the short term, I would sort of envision this as more of a companion, and that really relates to, kind of the time scales that, we're operating at versus, the time scales that CDISC operates at.
Yeah, and I'd also like to mention that in addition to the technical to the implementation guide, there's of course the FDA's technical conformance guide, and we can expect other regulatory health authorities providing their preferences for how SEND data sets should be delivered to them. So we can anticipate that at some day in the future there will be lots of different guidances of how SEND data sets would be prepared, and that's going to be an ugly and challenging situation. And I think strategically this group is positioned to be able to look across all of those guidances and supply a guidance that is to the industry to prepare data sets that are going to satisfy the needs of all of those other stakeholders, so that it could be the central place for additional implementation advice beyond what the CDISC issues themselves.
All right, thank you both. And I see now that we're at the top of the hour, so I will close out the Q&A session. But thank you everyone for submitting your questions and joining the discussion today. Again, we'd encourage you to visit our website to get more involved and sign up for mailing list updates as we announce new events and new forums for discussion. And I'll pass it back over to Gloria.
Thanks, Kelsey. Yes, I'll be posting the recording of this webinar, and you'll be able to access the recording, the PDFs, and we'll also send all those appropriate links out as well to everyone who registered. So thank you again for your time today, and have a great rest of your day.